Not here, I've learned. It's a great session so far. Yeah, I would like to talk to you about the work we do for in tool development for metagenomics, and this is work that was mostly performed by Martin Steiniger, who has left the group 6 months ago and now is working with Steven Salzberg in the US. And Milord also contributed quite a bit to the protein assembler that I'm going to talk about. And more recently, Ellie and Annika joined the project and also contributed to the newest code. So before I talk about protein level assembly, I wanted to give you a bit of an overview of the tools we have developed in the last 3 or 4 years. This is an older version of the same slide we saw in the previous talk. I just wanted to emphasize that we need to do something to get back to the precision that or to the sensitivity in annotation that we had when we were still using BLAST for function annotation. And that is obviously because of this enormous gain in throughput of high throughput sequencing. So the costs for computing by far now exceed sequencing costs, by the way, also of course storage of the information. And the bottleneck are still sequencing searches, so this is what made us start working on metagenomics data analysis. And the reason is of course that in a typical pipeline where you want to know what is in your metagenome, you have to do a lot of sequence searches after assembly to find out the community composition, so to get taxonomic labels, to get protein functions, to get pathways and so on. Since this is usually done not on just one, so to know who is there, what do they do and how do they do it, because you're not usually doing this on one sample, but on dozens or even hundreds, this is just very costly. So this was the reason for us to look into ways to speed up sequence searches while not really compromising on sensitivity, because a main problem in metagenomics, especially if you're going to more diverse metagenomes, let's say apart from gut, which is by far the best research, you get a problem of really low annotation depth. This can reach sometimes 90% of unannotated genes. So the goal was here to speed up the searches by a big factor and still get a sensitivity like BLAST or even better. So this tool is called MMSeq. I can't tell you really how it works, but this was published a bit less than 2 years ago in Nature Biotech. So this is just the summary of the main results. So here you have the sensitivity measured in a large benchmark, it's kind of AUC sensitivity for up to the first false positive, and in thousands of queries. And this is the speed up in comparison to, I don't see this dot really. This is the speed up in comparison to BLAST, and you see that MMSeq is here on the upper line of all these various tools that were available, some of the best are here, Diamond and Last, and you see that we are still quite a bit better. Thanks. Is it also responding here? No. So I have to use 2? No, I think it's okay. But what is really crucial advantage of MMSeq is that it is actually even more sensitive than PsyBlast, and PsyBlast uses profiles, so MMSeq can run with profile searches, and with the sensitivity that you see here, which is much larger than the one from BLAST, is achieved by doing a second iteration. So you do one iteration through a big reference database, you accumulate those sequences, and you do into a multiple sequence alignment, you do a second search, and with the second search, because you now have gained a lot of evolutionary information and put it into your profile, you can get much better, much more sensitive when you then do functional annotation. And this shows you the sensitivity and runtime for the second, third, and fourth iteration. So this second challenge that was pointed out 10 years ago was that because we are accumulating so many sequences, we are losing the capability to analyze them over whole environments, because we have nowadays thousands, even more, of data sets per environment. And one very central step of a compound analysis, of course, is to cluster the sequences to first get rid of all the identical or nearly identical ones. And because this requires quadratic runtime, you have to basically compare everybody with everybody. This is a major problem in the analyses if you want to go across all data sets of an environment. So the sequence clustering problem we addressed with a tool called LinClust that was published a year ago or so in Nature Communications. And I wanted to first explain to you why clustering is basically quadratic. So n is here the number of sequences that we put in. For example, assembled proteins that we call from assembled sequences, or this could be reads even. And k is the number of clusters that you will get out in the end. So this is the complexity that you know, of course, from k-means, but it's also the complexity of, for example, clustering with CD-hit. And so this is the algorithm that CD-hit and many other clustering tools use. So it's a greedy search. They take the first sequence. It's automatically a representative sequence because it's not similar to anything that you have already chosen as representative. Then you take the next sequence. If it's not similar enough, it founds a new cluster, and then you go on, and every time you find something that is already similar to something that you've already found, then you put it into that cluster, and if it's not similar to anything, you establish a new cluster. And obviously you have to do all the comparisons of all those sequences here with each one of those representatives. So it's basically half nk. And because k is usually on a similar order of magnitude as n, this is basically a quadratic problem. And you can see that when you look at the runtime of clustering for various data set sizes. So here linearly is the size of the data sets that we clustered with the available tools. And you see that these are mostly behaving quadratically or to the power of 1.8 or 1.7. And LinClust, as I will explain to you in a minute, is basically showing a linear behavior. So that means that you can cluster not only sets of 120 million where the current tools really start to fail, all of them, but you can cluster sets of really billions of sequences, at least a factor 100 more. So how does it work to go from a quadratic complexity to the linear one? So this is the way we came up with explaining this. So it's k-mer-based, like most other fast tools for sequence searches, alignment-free sequence searches. You have a step that is alignment-free and where you usually look for similar or identical k-mers. And this is the same in LinClust. So here we pick per sequence just a finite number of k-mers, let's say 20. That's actually the default value. So each dot now represents one k-mer in a sequence. So we have 20 times n such dots here, right? And the circles indicate which of those belong to the same k-mer. So these would be 4 sequences here that all have this green k-mer. And now we can pick one of these for each group, and I will show you in a minute how this works in Plus, how this is implemented in Plus. And we trivially take just the longest one of those, because then we know that we are not losing any information. We take the longest one, and then, and this is now important, we compare everybody in the green group, for example, with this longest sequence here. And everybody in the blue group also with the longest sequence, which happens in this case to be the same one here. So we do these comparisons, and because there are just n times 20 of these dots here, these are the k-mers in each of the sequences, because there are just n times 20 dots, and you just have one alignment per dot at maximum. You just need n times 20 alignments to check whether these sequences are actually similar enough that to keep this edge. So then what you don't do is you don't compare everybody with everybody. If you do that, then you are back at a quadratic algorithm. So as a last step, there's a trivial algorithm. For example, you could use set cover that will then take the graph where you have pruned away the edges that have not been verified or validated by your slow alignment algorithm. You prune them away, and then you do some simple clustering on the remaining graph. So that this works, I showed you on this slide here. This is very fast, and actually the sensitivity is similar to the best tools that were available a year ago. So we use this idea. That's also the reason why I wanted to explain to you how a linked cluster works. We use this idea for metagenomic assembly, but we're approaching metagenomic assembly from a bit unusual angle, because we are assembling protein sequences. So this is in a way a much simpler task, but that's also a reason why we are actually much better in getting protein sequences than if you first assemble on the nucleotide level, nucleotide sequences, and then predicting your proteins. And so this is what I wanted to explain to you now. So metagenomic assembly is, of course, a complicated task, as we know. So PLUS is the program, is the method that we developed, called protein level assembler. It's a complicated task because you have many genomes, usually hundreds, or actually for more complex genomes like soil or ocean, thousands of genomes, often with small coverage and also with population diversity. So not all the reads that come from the same species or even the same strain will have identical overlaps. And there are two approaches, general approaches for assembly. One is the earlier one, is called overlap graph approach, where every read is a node, and you connect them when you found a significant overlap between them, such that you believe they belong to the same genome. And because these overlaps can be 50 or 100 or so nucleotides or more, this is really relatively specific. So you can actually tolerate, let's say, 3-5% or so, maybe up to 5% of SNPs, of dissimilar nucleotides, because you're looking at a relatively broad, long overlap. The problem with these read-based assemblers, these overlap graph assemblers, as you probably know, is that they failed when Illumina came up because they need a lot of memory. Actually, each read has to be stored, which with Illumina was not possible anymore when it was introduced. And they are also very slow because they actually they scale with the read. So they basically have even if the coverage grows, there's no reduction factor, basically. And the idea of the Brewing graph assemblies was to gain this coverage factor by just representing k-mers, for example, 21 mers or 51 mers or so. So then if you have a coverage of 100, it doesn't matter. You present all those k-mers that are identical, those 100 k-mers with one single node, and this is the node that carries this k-mer. And then now the meaning of the connection is just whether these k-mers have an overlap, or basically whether they occurred in the same read. And then you in both cases you look for the shortest path and so on, and all this logic, of course, is quite well known and not very different between those two. The big drawback here, I mean, it's fast. It's much faster than this one. It's also memory. Saves memory, but it has this problem of best sensitivity to specificity trade-off. And this is crucial in metagenomics because you often have diversity in the populations. These are not clonal strains, these are wild strains, so you have similar strains, but even within the strains you probably have quite a bit of variation in many cases. And these lead to non-identical k-mers that basically produce bubbles and so on. Then gets very difficult for the graph to get interpreted into genomes, particularly because you need relatively long ones here to get significant and to be able to distinguish between one species and a closely related species. Right? So you have this problem of specificity sensitivity trade-off that was solved here, but actually it has disappeared here. And so the idea of PLUS is to have both of these advantages, to have a good specificity sensitivity trade-off by using real overlaps, but to calculate these overlaps not in quadratic time, basically, or on time coverage times n, but in linear time. And the second advantage that we have when we assemble on the protein level is that we're getting rid of most of the variation, actually, because most of the variation does not appear on the protein level because it's silent. That's, of course, a big advantage. Also, many of the repeats are actually either in intergenic regions, or they are not relevant for us because they are, for example, transposons, and they are completely separate from the proteins that we assemble. Plus, you've already heard how LINCLAS works. I explained to you how PLUS works now, and you will see that LINCLAS is basically a part of it now. So we take all the reads that we get from a metagenomics sample, we translate it in 6 frames automatically, so this is shown here with these arrows, and we're very greedy there, so we don't basically lose anything. And then we select in each of these translated protein sequences, these potential sequences, we select 20 or more chimeras. In this case, I just draw 2. And so 5 sequences, 2 chimeras, and then we sort them by so all of these 10 occurrences, so m times n occurrences, by the alphanumerically by the value of the chimer that represents the chimer. And then because of the sorting, we automatically have them sorted into groups of chimeras, right, of identical chimeras. And sorting is basically linear in time. It's n log n, and the log n is so small that it's basically linear. So once you have this sorted, you can now designate the longest sequence. So these are the starred ones. And then you sort again by the identity, by some identifier of these center sequences, which we call center sequences. And by sorting them, you actually bring together even groups that have the same center sequence. So in this case, for example, the pink one and the yellow one. And once you have those, now you compare everybody with the center sequence. So in this case, the center sequence is the star, so you do these alignments. There they are. You do the alignments with the star sequence, and you extend them whenever you find that the alignment is significant and satisfies your threshold that you've defined. And then we do this actually iteratively, so we go back again here, and then we try to extend even further, and we do this 12 iterations, at which point we see little improvement. So actually the title here is wrong, so just ignore it. We devised a benchmark that would allow us to show that the quality of these proteins that we assemble in a relatively greedy fashion is high. And so we took 2 very difficult benchmark sets. We took single cell sequenced genomes from Prochlorococcus and from a large number of diverse genomes, a good part of which is also Prochlorococcus. And we sampled reads from them. And then we try to assemble these reads with either PLUS or with other tools that are shown here, so MegaHit, Metaspace, and so on. And on these nucleotide assembled sequences, we then predicted proteins, and then we compared how many proteins do we get with the conventional approaches, nucleotides, and then predicting proteins, and how many do we get with PLUS. And so we show here for these 2 data sets, and I should maybe point out these data sets are very difficult because these cells are very, very similar. So it's really difficult to keep the different ones apart, and we thought that this is a good benchmark because this might be just the problem that PLUS has. If it's not very, very specific in these overlaps, it might make mistakes here. And you'll see that this is actually not so much of a concern at all. So we look at first the sensitivity and then the precision. The sensitivity of these assembled protein sequences is the fraction of reference protein sequences. So the references are from these reference genomes that match an assembled protein sequence with at least X% sequence identity, and this includes coverage. So if you match, let's say, 90%, it means that 90% of the residues of the reference protein must appear in one of the assembled proteins. And then you look at what is the fraction of this. So the higher, the better, obviously, and you see that PLUS, both with a 90% sequence identity, minimum identity in the overlap region, and with a 97% minimum identity in the overlap region, is quite a bit above the other assemblers. And then the precision, this is what is critical, of course, for PLUS, is the fraction of assembled sequences that match a reference sequence with at least X% sequence identity. So now it's the other way around. So if you make a lot of mistakes in these assemblies, you will not be able to match them to some reference sequence above a certain sequence identity. And indeed, you see that on this more difficult benchmark, where we have many diverse and also very similar sequences, you see that PLUS, particularly if you take a very aggressive 90% sequence identity threshold, it loses precision, above a sequence identity of 90%. The other version fares better here, but still is a bit below the reference tools. Now, interestingly, the question is, do these sequences that we assemble because of their lower precision, do they have a negative impact on function prediction? And the answer is clearly no, because they do have a lower precision, but they have a longer length because they're usually more complete. And if you look at what the quality of functional annotations is using eggnog, so we can run the eggnog mapper both on the assembled proteins and on the reference ones, and then we look at whether the annotation is the same, then you see that the false discovery rate for Plus is actually quite a bit lower than for all the other nucleotide tools. And this is just the specificity precision benchmark, but most importantly, this is the main result. Most importantly, when we now run Plus on big metagenomes, for example, on human gut samples, on the full set of eukaryotic transcriptomes from Tara, on one soil sample and on 11 soil samples, you see that the number of amino acids that we can assemble beyond a certain minimum length is in human gut is not much higher than megahit as a reference, but already in the marine microbiome is factor 2.5 larger, and on soil we get up to a factor 10 or more assembled proteins from the same data. And you see also that the length depends actually quite favorable, so we assemble a lot of long proteins. And the similar thing you can see depending on clustering threshold. So we are not just assembling variations, but when you reduce redundancy by clustering, you see that this factor really holds up. And so we use Plus to assemble huge data sets. So from 640 soil samples, we assembled a huge data set of proteins that we called soil reference catalog. From 293 million, we assembled also 293 million eukaryotic protein sequences from this Tara set, transcriptome set. And in an earlier paper, we clustered all proteins that we predicted on JGI contigs, and together this is now a big fat database, but that is by far the largest protein set available, and it's you can download it freely. And we just checked that what happens if we look at a famous protein that was discovered recently and published in a Nature article, a Cas effector that is much smaller than Cas9, and we looked at how many of those we find. In the original article, they found a couple dozen, and when we do the search in our big fact database, we actually find 190 of these new Cas14 effector proteins. And with that, I just want to, summarize very quickly. So we think that this is a very nice technique for getting catalogs of proteins for various environments. Some of them we've already generated, but there's a lot of other implications. For example, this richness of protein sequences is particularly interesting for people like us who do sequence searches, who want to predict structures using homology, using covariation, and so on. So all of this will be helped by this data. We are working on assembling viruses using a hybrid technique between protein and nucleotide level, switching back and forth. Yeah, and I want to thank you for listening and thank particularly the people involved in the work, so Martin, Milot, also Ellie and Annika. And there's a poster by Ellie which will also have these nice stickers. Maybe you're interested, so just drop by and you can get your set. Thanks a lot for listening.
Thank you very much. Does it work? Hello? Yeah. Time for one very quick question. Yeah.
Thank you for the talk. Interesting algorithms. How many of your proteins are full-length proteins, and how many of? Because you likely get lots of fragments.
Yeah, that's an interesting question. It's very difficult to say because for many of the proteins, we don't really know where the start codon is. So what we did is we looked at where we are sure that the start codon must be there, because before we have a stop codon, we sequence the stop codon directly before, and we've seen basically the start of the protein alignment. And this is something like 15% or so typically in these data sets. But probably it's more. But length is, of course, a problem is particularly in very diverse data sets like soil. So it could be that there we have actually a higher fraction. I think the average length is only 200. So it could be that a higher fraction there is not complete.
Okay,
Thanks.
thank you very much.